Hello, everybody. Today I'm here to talk about DNA replication as well as DNA structure. As we know, DNA is a molecule made up of two strands twisted around each other in a double helix shape. Each strand is made of a sequence of four chemical bases represented by the letters A, C, G, and T. The two strands are complementary to each other, which means wherever there's a T in one strand, there will be an A on the opposite strand, and wherever there's a C, there will be a G on the other strand. It looks a lot like a ladder, and it has a 5' end and a 3' end, as it's known. The two strands separately, or as they're connected, they run in opposite directions, and this is how DNA gets replicated. The first step into DNA replication is to separate the two strands. The unzipping is done by an enzyme called helicase and results in the formation of a replication fork, much like a fork in the road. The separated strands each provide a template for creating a new strand of DNA, and another enzyme called primase begins this process. This enzyme makes a small piece of RNA called a primer. The primer is the starting point of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction. As we mentioned before, it has a 5' end and a 3' end. DNA polymerase can only go from the 5' end to the 3' end. And once one of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adds bases one by one in the 5' to 3' direction, while the other strand, known as the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. So the DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and process is completed over and over again. Once a new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind of the DNA. And finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double-strand DNA replication, as is described as semi-conservative because each DNA molecule is made up of one old conserved strand as well as one brand new strand. Thank you.